Okay, good evening, everyone. Do a quick audio check here. Can you hear me online? Talking to John. Yes, we're streaming. Great. Well, thank you very much. My name is Steve Gotch, and I currently serve as the Canadian chair of the Northern Fund Committee. I'd like you to welcome you to our special session that we're hosting here this evening. Tonight's presentation is part of an initiative that's been undertaken by the Northern Fund Committee to showcase projects and initiatives that have received funding from the Northern Fund to advance implementation and cooperation through the Pacific Salmon Treaty process. A little bit about the Northern Fund Committee itself. The committee is chaired by myself and Commissioner Doug Vincent Lang from Alaska, who's in the room today, as well as supported by members Commissioner John McCullough, Commissioner Russ Jones from Canada, and then from the United States, Commissioner Bill Auger and Mr. Jamal Moss. Over the past 20 years, the Northern Fund has contributed over $83 million US to nearly 800 projects. So the $83 million figure is significant, in particular when we consider that the original capitalization of this fund was $75 million. So we've actually put more money out the door than was originally invested, and we continue to disperse around $5 to $6 million US each year. Tonight, we'll be placing the spotlight on, in fact, a series of programs and initiatives that have been funded by the Northern Fund, focused on assessment and research into Canadian origin salmon stocks in the Taku River watershed. Taku River programs are unique, not just in a Pacific Salmon Treaty context, but also nationally. These programs are one of a few examples of projects which are truly co-delivered through bilateral cooperation between Canada and the United States. And so, without further ado, we have 4 presenters that will be leading our session this evening. From the Alaska Department of Fish and Game, we have Jeff Williams and Flip Pryor. And from Fisheries and Oceans Canada, Aaron Foos and Corino Salomi. So please welcome our presenters.
All right, well, good late afternoon, everybody. It's great to see so many people here. My name is Jeff Williams. And as Steve mentioned, I work for the Alaska Department of Fish and Game based out of the Douglas office. I'm very privileged to be here in front of everybody to have the opportunity to talk about Taku River salmon stock assessment and enhancement work. The program has an amazing legacy of cooperative program delivery. We wanted to start the presentation a little different, knowing that there's been so many people that have worked for years, decades to get these programs where they are today, on both sides of the border to get implementation and develop these programs. So we're going to start by thanking all the people that have involved over the decades. I mean, I would have to guess maybe over 1,000 people. And prep for this presentation, I wrote all the names of the people that I've worked with from Taku River Tlingit First Nation, Alaska Department of Fish and Games, and Fisheries and Oceans Canada, just near the border. And I came up with well over 100 names, over 120 names, just folks that I've worked with in one section of the river. So you expand it out over decades. It's not countless, but it sure is a lot of people. So we want to acknowledge them. We also know, like everybody in this room, I'm sure, that doing remote salmon stock assessment work and enhancement work is not a cheap endeavor. And so we'd also like to thank all the funding support given by numerous sources over the decades as well. And the Northern Endowment Fund has been a large contributor since the beginning. We've received funds on the Taku River in one way or another since the first year in 2004, I believe, Steve. So I'd like to thank all our funding sources as well. So with that, we'll get into the presentation. so this is very, very cooperative work. So the presentation is going to work very similar where we're going to be doing a lot of back and forth. So.
All right, thanks, Jeff. Aaron for the Salmon Fisheries and Oceans Canada. Again, very privileged to work on the Taku River for the last 8 years in my case, but before that with many of the partners. And many thanks to the Northern Fund for extensive funding and support to a lot of our ongoing programs. As you've probably picked up by now, it's very extensive programming. We do a lot of work on the Taku across a number of species. The Northern Fund funds have touched on almost all aspects of that. It's very hard to compile that into a short talk. So we're kind of giving an outline. So here's the outline of our outline. We'll work through it quickly. We'll orient you to the Taku watershed and where it is, some basic information. We'll talk about the history and land use history. Jeff alluded to a long time series of salmon assessment history, so we'll give a bit of information on that. And then we'll get into a bit more of our actual stock assessment work that we do. We'll just be touching very high level on almost all of these things. We'll work through it in sort of an annual cycle approach in the way the salmon utilize the river. So starting with smolt work early in the year, working through the species of Chinook, sockeye, and coho into the river. It's not possible to do it entirely chronologically, but we'll do our best to stick to that approach. And then following our stock assessment, we'll turn it over to Flip and Corino to talk about the enhancement projects that are also occurring on the Taku River. So where is the Taku River? The Taku River is in northwest BC and southeast Alaska. Sits right up there in the corner. We, due to the treaty, we focus on 3 main species, Chinook, sockeye, and coho salmon. Those are the key salmon species. If we zoom in a little bit more to the Taku watershed, get the lay of the land, about 90% of the watershed is in Canada. It drains out into southeast Alaska. The red line is the international border. And you can see the city of Juneau in southeast Alaska near the mouth in the marine environment. And the nearest Canadian community is right up there in the top corner, Atlin, British Columbia. And that's the primary jumping off point to the Taku drainage from Canada. One thing that's really unique about the Taku River is its incredibly pristine drainage. There's very little development. There are no roads in the entire drainage. There's been a little bit of mineral exploration work, but there's been no major mines. There's been no major hydro. There's no major forestry operations. So it's largely a pristine watershed. A lot of consideration is given, and it's considered a salmon stronghold by the First Nations and many of the partner agencies that are working in and around the Taku. So it's quite a privilege to work in a place that is still so pristine. That also makes it a challenge, as Jeff noted, because all of our access is required by air or boat to the lower river from the marine environment. So it's very costly. Moving into just touching on the distribution of the salmon. This is a little bit out of sequence, but gives a sense of the salmon utilization of the watershed, that almost the entire drainage is available to anadromous fish. There's only a few upper reaches that are impenetrable, or there's a canyon on a couple of the systems, but you can see, don't worry too much about the detail on here, but the dark highlighted lines show the distribution of the 3 key species we're talking about throughout the drainage. This information came through telemetry projects. There were telemetry projects in the 70s, but a series of more recent ones, 2015 through to 2022, we did a number of years of telemetry based on all 3 species in the Taku to learn distribution and inform these maps, but also to inform some of our stock assessment work in terms of what our biases from handling mortality was in our stock assessment programs that we'll be talking about. So informing tagged fish dropout rates. We'll touch on that in a few later slides. So moving on to the history of the Taku. Obviously, it's been inhabited by indigenous peoples for thousands of years, long history of use. The Tlingit First Nations primarily in the main stem part of the Taku, both coastal and inland Tlinkits. They've been living and stewarding the drainage for time immemorial. Obviously, the abundant salmon runs were a critical piece of that. So a lot of the human habitation and settlements were all focused around areas of salmon abundance and access to salmon at key times of years. The Taku is also a bit of a trade route by foot, one of a few trade routes to the coast mountains. So a lot of passage through there, but unlike most of the others, it never turned into a trail, wagon road, highway, etc. It's remained remote and largely impassable. The Taku River Tlingit First Nation out of Atlin in BC is the entire Taku drainage is their traditional territory. We work extensively with them through not only the treaty process, but other processes as well. And the Tahltan nations out of East Lake have traditional territories in the upper parts of the drainage and overlap areas, so we collaborate with them as well. Both First Nations are heavily involved in the treaty process through our technical committees, but also have very well-developed fisheries programs, are fully engaged and collaborative in all of our TTC and stock assessment enhancement processes. And both nations are I would consider quite leaders in terms of salmon conservation, knowledge and research into salmon utilization in the drainage, and heavily involved in management and our stock assessment, as you'll see moving forward. And Jeff's going to talk about the sport.
All right, continuing out toward the marine environment, the estuary and inlet. There's been use documented for well over a century. The sport fish records are not totally clear from what I've found as far as actual harvest numbers, but it's pretty clear that there's been sport fishery or people targeting Taku River Chinook, at least, and sport fisheries for well over a century. And moving to commercial fisheries, the records go back a little further, and there's documented harvest for the drift gillnet and troll fisheries targeting Taku River Chinook and sockeye, chum, and coho going back to 1904. So a lot of use history for many fisheries. I really like this picture. It's in the lower river in the estuary, essentially. There's these tenders lined up, and a lot of that's filled in with silt from the glaciers over the years. And those boats would probably be sitting on land today, potentially. See Chris nodding there. Yeah, pretty cool shot. A little more history about stock assessment. Well, it was before statehood that Alaska started doing government-led stock assessment for Chinook salmon. This work started right around 1950 to 51, and there's records going back that this stock assessment information was shared with the Canadian government from the very beginning. So we've got collaboration from the very beginning, going back at least to the 50s. Kind of moving forward to the signing of the treaty in 1985, a lot of the projects that are in the water today started around this time and continue today. These projects got more refined with the 99 agreement, and with that was the chapter 1, the Transboundary chapter, where we spend a lot of our lives. And like we've mentioned before, we've seen some kind of funding through the Northern Endowment Fund since 2004, since the first year of awarding funds. All right, looking at why we do what we do, just a little background and context. So looking at chapter 1, the Transboundary chapter. Object is to have viable fisheries by both countries, and the intent in the chapter is to have abundance-based management for Chinook, sockeye, and coho salmon. Each country has harvest shares, so there are directed fisheries on all these stocks. Also, harvest shares for sockeye salmon are based on the wild and enhanced fish proportions. And what does stock assessment do? Well, throughout the season, we provide in-season estimates of abundance that's used to manage these fisheries sustainably and to meet treaty obligations. Kind of moving a little closer to what we do each season. So the chapter prescribes a technical committee that all four of us that will talk today are on. And each preseason we meet and we talk about the plan and how we're going to cooperatively lay out the stock assessment work each season, which is the segue into the stock assessment work. So this is just a quick picture of what fish will look like from the banks of Canyon Island in 1951. And we have a fish wheel there still in 2025. There's been some breaks, but overall we've shows a long history of some somewhat consistent effort over the years, which is just pretty cool to see. Sorry. So the first project that we start out each season doing in April is the smolt tagging project. So this project focuses specifically on Chinook and coho salmon. It's a 2-month project, and it's got a joint crew between TRTFN, ADF&G, and DFO. These fish are marked externally with the adipose fin, and they are tagged with a coated wire tag. I'm sure a lot of people in the room are very familiar with the coated wire tag, but if you're not, it's a piece of metal wire with a unique coat on it, and it's just over 1 millimeter long and about the width of a strand of hair. So the crew goes out to capture these fish using baited minnow traps and beach seines. We've got 3 trap lines each spring. One of them is completely below the US-Canada border. The other one straddles the border, and the third one is above the border. And the crew go out each day, capture fish, and bring them back to camp. And we also use beach seines to capture, and they're kind of broken up. One half of the season is focused on fish that are holding and haven't started to migrate out yet. And once fish start to migrate out around the first week of May, the crew switches gear and starts using beach seines to capture the majority of the fish. And just a quick look at what a beach seine looks like on a shallow gravel bar. So what do we do with all these captured fish? So they all come back to a central location. The crews bring them back to a floating tag shack that you can see on the left. There's some live net pins that hang off of the net, so the fish will sit in those pens until they're processed and go back in the pens overnight to check for tag retention. The photo on the right is the crew processing fish. So all the fish that are captured need to come up to a table. Bycatch, or exotics as the crew calls them, get weeded out, species that are not going to be tagged, and then all the healthy Chinook and coho move down the line to the tagging machine. Here's just a quick look at what a tagger looks like. So the larger picture on the left is someone holding Chinook smolt upside down into a head mold, and each individual fish has to get ran through the machine to be tagged. So it's a very labor-intensive project between both capture and tagging. And the second, or the 2 pictures on the right just kind of show a zoomed-in shot. Yeah, you could see the tag on that screen. Just the placement of the CWT in the snout of the smolt. So yeah, we're going through these projects quick because we have a lot to cover. So just kind of looking at the number of smolt tagged on the project going back in time. So the first year of tagging for coho was 91. The projects have continued through the years. Chinook started in 93, and the crews over the decades have put out almost 1.7 million coated wire tags. So tremendous amount of work to go out in the river and catch these wild fish and bring them back and process and tag them. So just, yeah, quick look at some of the results and just showing the time series that these programs have been able to produce over the years. So The black bars you see are the smolt abundance estimates. So we're estimating wild smolt that leave the river every year. Fairly unique for wild systems. And then that dashed line is the marine survival of those outmigrating fish every year. And a similar figure here for Chinook. The only difference being the x-axis, instead of being a calendar year release, it's tallied up by brood year, and that's brood year of fish that return at ocean age 3 and older. And so, yeah, the average for, oh, sorry, going back one slide for coho, for those are interested. It's roughly 10% for the long-term time frame. We've definitely seen some really low marine survivals in recent year, which has led to some low adult returns. Actually, some positive news on the Chinook slide. You could see that line in recent years is ticking up, and the long-term average for Chinook marine survival is roughly 10% for Taku Chinook. We saw a lot of years in recent history where it was a fraction of a percent, so it's nice seeing the incline or the increase in marine survival, and we're seeing the increase in our adult returns now because of this. Oh, sorry. So for coho, this slide right here. Yeah. Yeah, sorry, going back and forth. So that's 10%. And for Chinook, it's closer to like 2%, 2 to 3. All right, moving quickly here. So we're going to take a look at Event 1 tagging for the adult mark recapture programs. So we tag just below the US-Canada border each year from April through September. We tag for the mark recaptures again, Chinook, sockeye, and coho salmon. And we use two types of gear to capture fish for the mark recapture, one being a drift gill net and the other being fish wheels. And we have 2 of them. This picture right in the center is Canyon Island. You can see how the whole river chokes down, and that's where fish wheels spin in that canyon. It's a very unique spot that allows fish wheels to spin without being wiped out by debris. Too often it does happen. So looking a little closely at the drift gill net. So this is a 2-month project specific to capturing and tagging adult Chinook salmon. We set a net out, we record times on each drift, and as soon as the corks start bobbing, the whole net comes in. Fish get cut out of the net as soon as possible and placed in a tote to sample. All the fish are sampled for the presence and absence of an adipose fin. So our event 1 for the adult mark recapture is our event 2 for our juvenile project. So looking for the presence and absence of adipose fins lets us know if the fish was tagged as a smolt as it outmigrated. We also sample these fish for age, sex, and length information. And we use a tag called a spaghetti tag for the primary tag for the mark recapture. We'll take a quick look. So the picture on the left, the larger circle just behind the dorsal fin of this fish is a spaghetti tag. The tag is ran through the dorsal musculature of the fish and has a unique tag, so we know the specific time and date that the fish was released, as well as all the biological information collected with it. You could also see on this picture on the left, it shows a fish with an adipose fin. That fish was not marked as a smolt. In contrast, the photo on the right shows a fish that is clearly missing its adipose fin. We generally sacrifice these fish and send the heads to our labs so we can determine exactly when that fish was tagged. All right, the second gear type for event 1 capture is fish wheels. We have 2 fish wheels, like I mentioned, in the canyon. They spin for a consistent amount of time each day. Once this gear goes in the water, it's fished every day for 5 months. So all Chinook and coho and the fish wheels, again, are sampled for the presence and absence of an adipose fin, which gives us the event 2 information for our smolt project. As well as being sampled for the presence of adipose fin. Chinook, sockeye, and coho, and the fish wheels are sampled for age, sex, and length and tagged for the marker capture. All right, so that's moving through event 1, and Aaron's going to take us through some event 2.
Thank you, Jeff. So as Jeff mentioned, just on the US side of the border, they're a few kilometers below the international border at Canyon Island. They apply the marks for the marker capture. We use Canadian commercial and assessment fisheries just above the border as the recapture or event 2 component of the marker capture. Just draw your attention to the map there, kind of in the center. I know it's small, but the key piece is you can see the international border transecting the river, and the gray highlighted section upstream of the border. That's the Canadian fishery area. It runs for 18 kilometers upstream of the border. A majority of the fishing happens in 3 or 4 kilometers just above the border. So our event 1 and event 2s are very close together, which is very beneficial from a marker capture perspective and helps us with a lot of our assumptions around tag loss and those types of things. Depending on the species, we're looking at 2 to 4 days to transit between the 2 events. The picture in the lower end is just representative of our commercial fishery. That's Chris Kendall, who's here in the room, and his wife Amy. The fishery is small scale. It's typically conducted from 3 to 4 meter skiffs, short gill nets that are drifted generally within the river, as well as set nets. All the nets are pulled by hand and fish sampled by hand in small crews. There's 19 commercial licenses on the river, but typically we have somewhere in the range of an average of 5 to 9 licenses fishing, depending on the year and the season. So pretty small fishery, but the key part of the fishery from stock assessment is that during these directed fisheries, which happen on returning salmon, depending of course on abundance and our information available, Chinook haven't had directed commercial fisheries for over a decade now. So we'll get into Chinook in a different manner moving forward, but sockeye and coho have consistently had directed fisheries. We use those fisheries as the recovery, as I noted, and it's a condition of license for fishers to inspect fish for marks, the tags that Jeff just showed us, and to return them to DFO. So they also are required to inspect for adipose clips, which informs the smolt marker capture, as Jeff alluded to. There is allowances for assessment fisheries under Chapter 1 in the treaty as well, so it's at the direction of the panel. In some years where we've determined that the directed commercial fisheries aren't going to be appropriate or adequate, there is the ability to implement assessment fisheries to support the marker capture programs in the Taku. So from a sampling perspective, our Canadian crews are on the lower river interacting with the commercial fishery on a daily basis. As I mentioned, the active fishers submit their harvest data to our crews who collate that, which because it's a condition of license to inspect for tags, that gives us our inspected number for our marker capture. It's essentially the entire commercial catch or assessment fishery catch. And then the tags that are turned in are obviously our recoveries. Our staff collect those each day, as well as go through the harvest and do subsampling. We do all the biological metrics you would expect on these fish. We look for tag scars and secondary marks just to ensure compliance and tag loss and all those good marker capture activities we do. Our fishers are extremely good at providing this information, but we do sometimes have a small correction factor, but we're able to generate that from our inspections. Another key piece of what we do is otolith recovery. We have enhancement programs, which we'll hear details about in a minute. Those fish are all thermally marked. And so we sample sockeye heads to determine the wild enhanced breakout of the harvest. So all that information that Jeff's crews on the Alaskan side and our crews on the Canadian side collect are shared on a daily basis. Like we're extremely integrated in terms of how we share information through the TTC processes. Every day, the crew on the lower river sends out full details on all the fish they've captured, sampled, tagged, all that relevant information, their fishing efforts. And likewise, on a weekly basis on the Canadian side, we send out all of our fishery effort information and then all our inspected and recovered information for the marker capture. So that provides information for managers, not in terms of what's occurring with the fishery, but also helps us collaborate on our stock assessment. So lots of data goes back and forth on a daily basis. Weekly, Jeff and I collaborate to come up with the estimate for the species of interest for whatever time period we're working in. And we generate a weekly abundance estimate from the marker capture data. We then can use projections to provide information on what we anticipate the escapement will be and whether we're on track to meet escapement, and then terminal run size estimates, which are provide the information under chapter 1 and set the allowable catch that's available. The the wild enhanced information we gather from otoliths also feeds into that to help inform harvest share, because in chapter 1 for sockeye, the enhanced component determines the harvest share between the two parties. This information also provides us for the postseason estimates for sockeye and coho. So that's fully done through the commercial fishery. Postseason, we review all the data, finalize all our estimates. And we have, as I mentioned early on, information on the bias from our handling of fish. Jeff talked about the fish wheels and the gill net capture. So there is some mortality of fish that we're marking, and we've been able to quantify that. So we incorporate that into our estimates. So we remove that bias. We do that for sockeye and Chinook. But we will talk about Chinook postseason being done a little bit differently in coming slides. We also have the ability to do time and size stratified estimates where those are appropriate, looking at size distribution between the events and all the details around, the spatial distribution of catches and all that fun math stuff we get to do around marker capture. So Chinook, as I mentioned, are a little bit different. We haven't had commercial or assessment fisheries for a number of years on the lower river. So our event 2 or recovery episodes for Chinook happens in the headwaters and up on the spawning grounds. But one key project before we get there for Chinook is the inriver sonar project. This one gets special mention because it's been entirely funded by the Northern Fund for its 9-year existence. It started as a assessment project just to see if we could do enumeration with sonar at this location. It's turned out to work fairly well, and working with some contractors, we've devised a pretty reliable assessment method using this. So much appreciation for this specific project to the Northern Fund. I mean, everything we've talked about is funded, but this one has been exclusively funded by them. It allows us to enumerate 3 of our key Chinook populations into the upper drainage, which from a Canadian perspective also comprise an individual conservation unit. So very critical information. It also gives us an early look at Chinook abundance into the upper river. And Jeff.
All right, taking another look at Chinook abundance up in the spawning grounds. So we've have an aerial program for Chinook salmon specifically. This is the longest running continuous project we have for any species on the Taku. Methods and survey areas have been standardized since the early 70s. These surveys consist of flying a helicopter with the door off at low elevation. And we surveyed 5 of the most abundant spawning reaches in the drainage. We have ran this concurrently with multiple years of mark recapture, which has allowed us to generate an expansion factor. So we sum up the peak aerial counts across all the spawning tributaries, and we're able to expand that out for a drainage-wide estimate. This is an important project, especially on years of Chinook low abundance. Oftentimes, if we're unable to get a valid mark recapture estimate because of sample sizes, we've relied on this aerial program to provide us a valid estimate. which estimating escapement in the end is arguably the most important thing we do for adult Chinook on an annual basis. So again, kind of wrapping up the spawning ground work specifically related to Chinook. Like Aaron had talked about, the event 2 for the marker captured estimated adult sockeye and coho salmon takes place in the lower river, where for Chinook salmon specifically, it takes place across all the spawning reaches or main spawning reaches, 6 of them in total. So joint crews between Taku River, Tlingit First Nation, Fisheries and Oceans Canada, and ADF&G go out together on many of these spawning reaches. We mingle up in Canada because we're coming from different towns and find ourselves at the same camp sampling. And yeah, we sample, like I mentioned, 6 of the most abundant spawning reaches in the system, and multiple different gear types are used depending on the system. A lot of samples are taken from rod and reel. There's multiple weirs across the system. There's carcass pickups. We use several different gear types depending on the situation and the system specifically, as well as run timing and what stage in spawning the fish are. So once all this information is combined with our lower river event 1 tagging, this enables us to generate a postseason estimate of escapement on an annual basis. And like Aaron mentioned, like the other species, we apply a dropout rate that was learned from the radio telemetry work to reduce bias in that estimate each year.
All right. So just touching on a couple other aspects of our stock assessment. Like you said, we're running tight on time and can hardly touch what we do. So we do enumerate sockeye into 4 of our key headwater lakes in the Taku River. So a combination of traditional sockeye weirs as well as some video weirs. A couple of them are run by First Nations and a couple are run by DFO. These are really important and allow us to enumerate these key stocks, but they also serve some other purposes. There's some related passage monitoring. They allow us to see if the timing of fish is on or not, or if there's passage issues, because a couple of these populations have experienced passage challenges over the years. And the Northern Fund has touched on that by providing funding over many years, particularly through the Taku River Tlingit First Nation, to monitor passage. So these weirs provide information on that, as well as monitoring for the success of some of these passage improvement projects that they've been working on. So appreciate that involvement. And then these are also used to collect broodstock for the enhancement activities at 2 of these lakes, which Corino and Foot will talk about in a moment. Another project, or it's a vast area of our work, is the genetic side of our work. The Northern Fund started funding right when genetics became more mainstream, and so has provided extensive funding to develop our bilateral baselines in the Taku. Lots of money for the baseline collection of all of our species, and then ongoing funding to do the GSI work and any additional work that's happened around that. So special nod to the Northern Fund around our genetic work. It's a very bilateral process, again, just like all of our other projects. It's kind of hard to see, but in the far right slide there on the ground, you can see 2 bottles with their lids off. That's the way the sampling's done. We have an American bottle and a Canadian bottle, and so tissue sample goes into each one and they go off to each lab. So the collaboration starts right on the ground when we have our hands on the fish. And that's ultimately resulted in current SNP baselines that are coast-wide and bilateral for Chinook, sockeye, and we're currently working on coho in the Taku. And obviously, most people are aware of what that enables, but it specifically in a Taku perspective, and again, largely funded by the Northern Fund, has been stock comp of the harvest over some of the years, and then stock comp of some of our in-river runs through the fish wheels and some of those additional work we've done, as well as any other genetic interests we have. So that's another key piece. So Jeff's going to run through some quick results.
All right, just to wrap up the adult mark recapture. Yeah, so you could see a very long time series, continuous time series of information going back here. First look at Chinook salmon. I guess I don't really need to get into too much detail about them, but I will say the stack bars, the darker bars, escapement, and the lighter bar is the harvest. And just looking all the way back in time, we weren't able to identify a specific Chinook harvest in the marine fisheries until the coded wire tag projects had come around. So you could see that kicking up in the 90s when the program started. Taking a similar look at sockeye. Sockeye does have harvest. They've used different approaches, including scale pattern analysis, to estimate marine harvest in the terminal fisheries, and has been using GSI for many years now. But there's continuous time series with harvest in the marine fisheries. And also taking a similar look at coho over the years. Yeah, I won't get into any specifics, but folks could reference them if they want to, looking at the presentation at another time. And with that, we'll turn it over to our enhancement colleagues, and hopefully we didn't take too much time from you.
All right. I'm Pryor. I'm the aquaculture section chief of Alaska Department of Fish and Game. For treaty purposes, I'm the U.S. co-chair of the enhancement subcommittee, and I've been on the enhancement subcommittee since I think 2004. So I'm kind of a graybeard. All right, getting into the treaty in 1985 was first signed with the idea of collaboration in the management of the Transboundary Rivers. In 1986 and 87, there was disagreements that mostly were related to harvest shares. And so 1988, there was a new agreement which revised the harvest shares based on this idea that we would produce an extra 100,000 returning adult sockeye salmon to the Taku River system. In 1990, we began stocking projects at Tatsamini and Trapper Lake. Hit the right button and everything. Northern Fund has been very key to getting these projects done directly with the hatchery projects themselves, but also we've had feasibility studies on releases or possible release sites. A lot of them are not ideal, but we had to go through the motions and look at them and look at pros and cons. And then there's also been some salmon migration improvements, as well as I'll get into it later with the hatchery itself, but some infrastructure type projects. And then we're tag teaming fast here.
And there's only 10 more slides left. So did you get questions ready? Most people in here are probably fairly familiar with hatchery programs, but I just wanted to touch a little bit on the transboundary enhancement projects because they are, I think, very unique. A big remote landscape with a transboundary scenario. The adult salmon we use for enhancement are captured at a stream or lake of origin, typically by net or off of a weir, like the ones that Aaron talked about. The eggs and milk are collected in the field. The eggs are fertilized in the field. They're disinfected in the field, and then they are flown to Snettisham Hatchery in Alaska near Juneau. So, fairly unique activity there. The eggs are incubated in quite cold, fish-free water off a mountaintop. They spend the winter at the hatchery. They hatch, and then as quickly as possible, once ice out occurs on lakes in BC, the resulting fry are flown back to their lake of origin. And the photo of the hand there is in there to highlight that the fish we're sending back, the fry we're sending back, they're basically at 1/4 of a gram. So they're basically largely unfed for the most part, or minimally fed, just waiting for the ice out to occur.
And aside from a little bit of extended rearing that we do at Tatsumini Lake, that's it. So a relatively light touch enhancement project. And I put a red circle there to show that we're just touching on a bit of the adult return to feed the hatchery program. And then immediately after fry emergence, we're kind of putting these fish back into their waters of origin. And a lot of other hatchery programs, you might have 18 months of incubation and rearing in a hatchery with lots of feeding. And that's not the project here. Yeah. Lakes that we've been doing enhancement on, Trapper Lake, we've been using relatively small releases of fish into Trapper Lake to assess fish passage opportunities into the lake. There is a waterfall there that does limit sockeye migration. The lake does have some smolt production capacity, and so the fry releases we've been doing there have been helping us test that lake productivity, adult passage feasibility, and contributing a little bit to harvest. King Salmon Lake, Northern Endowment funding there has helped with some of the fish habitat and migration assessment, and then also 2 years of quite successful enhanced fry releases, smolt monitoring, and then subsequent adult returns. We haven't continued with that project, but we demonstrated that fry releases there do have the capacity to improve adult production there. I think I'll keep going here, right? Yeah, okay. And then I'll go into a little bit more detail now on Tatsumini Lake. So Tatsumini Lake initially had a direct fry release, but we tried, our predecessors tried to some extended rearing projects at the lake through 1997 to 2002, and then again in 2007. They weren't very successful, and the Northern Endowment Fund helped with a review of that program and resulted in some recommendations. And that project evolved over the years. Initially involved a fairly unique project with gravity-fed capilano troughs on the shore of Tatsumini Lake, where fry were fed for a period before released to the lake. That was advanced to then include some net pen rearing with various trials. The water supply to the cap troughs actually was washed away, and so more recently we've moved to strictly in-lake rearing, extended rearing for a portion of the fry that are brought to the lake. And more recently now we've been implementing about a 3 to 4 week short feeding period, and we bring those quarter-gram fry up to about 1.5 grams. So again, a fairly limited project. We're just trying to give those fish a boost to take advantage of the large Tatsumini Lake production potential.
All right. And yeah, this is a rather busy looking graph, but what it shows is the black bars are direct release into the lake, and the little dash bars are various enhancement projects. The darker looking ones are the capilano troughs, which we go back. The top picture there is the capilano troughs. The lower ones is the net pens. So the basic takeaway here is through the extended rearing, we can double the marine survival, but we are very limited in what we can produce in the lake due to there's just a limited number of places we can put pens. And yeah, the infrastructure up there is kind of limited. All right, we jump there.
All right, so Flip mentioned that we do actually produce adult sockeye with our results, and this graph here shows the white bar shows the naturally spawned adult returns to fisheries in a terminal run sense for Tatsumini Lake, and the blue represents the enhanced component of the adult run size. we did talk about 100,000 as being kind of a watershed wide target at one point for Taku. Obviously, Tatsumini Lake's not doing that on its own, but in some years there's been over 20,000 adult sockeye produced from our efforts. Next graph here shows the post-fishery, post-migration adult returns rate to Tatsumini Lake weir. And I think the point here is that there's a number of years where returns to the lake have been pretty small. And if you look at just the white portion of the graph, there's several years where there's less than a couple thousand fish. But in those years, our enhancement efforts actually almost doubled the overall return. And so, on one hand, we were trying to make more fish for fisheries, but I think here it's also showing that in some of the low years, we've actually been able to significantly increase the returns. And then 4 and 5 years later, that's helped to bring the run back up in size a bit. So a sort of mixed range of benefits from the enhancement.
All right, and this slide is showing Snettisham Hatchery. Snettisham Hatchery is a state-owned facility that is run by Douglas Island Pink and Chum Incorporated, or DIPAC. It's a very unique hatchery in that it's set up with a lot of isolation units. So we're running 2 different stocks. We got the Snettisham Hatchery stock, and then the TBR stocks are run out of the same hatchery. They have isolated areas and different water patterns, so everything can be biosecure between the 2 stocks. Northern Fund has been helpful in contributing in some of the infrastructure at the hatchery. One of the things is recently done was we put a chiller in. We call it DVL for no apparent reason. And it could be used to control the water so we could have the time when the fish come out, but it also can be used to help put otolith marks on the fish. And I'm assuming some people are familiar with these, but an otolith mark, so there's a photo there is an ear bone of the fish, and you can put those bands on the fish by controlling the water temperature. It takes about a 4-degree or higher change in water temperature relatively quickly, and it will lay those patterns down. Each project gets the pattern ahead of time, and through manipulation of the water temperature, we can put down these marks. So at various points of the fish's life, you can take that, you have to kill the fish, but take the ear bone, split it, burn it, put it under a microscope, and you see that pattern, and you can know the brood year of the fish, the release location, and in some cases, the various rearing strategies that were involved in that fish's life.
Yep, I think so. Yeah, you or me, whatever. I think Aaron talked about enumeration. So one of the other things that we're doing at Tatsumini Lake is smolt enumeration. So there is a mark recapture program that a contractor, Brian Mercer, does for us. Smolts are sampled. They get a polymer injection, different colors in the lake, and then they are released again and then sampled near the outlet. And that information is used to give a smolt out-migration estimate. We're also using the otolith marks to then sample some of the fish and get an idea about naturally spawned smolt production, direct release hatchery smolt production, and then our extended rearing smolt production. We also this year had NEF support a trial multi-beam sonar program. So instead of using the mark recapture, we have a sonar at the lake outlet to provide potentially an alternate or additional method of enumeration. And this project's basically going on for about 27 years now. So a pretty long data set for these smolts. And just wrapping things up, some closing points. So, mentioned that the enhancement project, I think, is a relatively low intervention program. It's definitely increased harvest abundance and opportunity for harvest. The program's unique. We can do in-season review of returning sockeye to the lakes and make adjustments in our enhancement, and in some years help to augment low brood years. It's not magic though. Enhancement's not a silver bullet, particularly in this watershed. It can be logistically challenging, and there's a cost, and there's always a risk to enhancement that you have to manage. They are manageable, but there are risks there. And we haven't met the original goal of 100,000 watershed wide, but I think we've done some decent work on the lakes where we have done the work. The accurate hatchery fry releases we do have a good count of the fry releases. Provides us with a baseline of fry in a lake, and then later on when we do the smolt enumeration, it gives us information on lake productivity, and it allows us to compare what we might expect from naturally spawned fry and smolts. We do have a really long-term biological sampling program. We have lake limnology, we have salmon age, sex, and length, natural pathogen profiles from the sampling we do, and maybe somewhat uniquely a long-standing fecundity information as well. Overall, the work we're doing in transboundary with enhanced fish is really helping us to understand lake productivity and potentially climate-related influences on lake productivity. And we have been trying to update our limnology work. We've been working with Cultus Lake Lab to get some improvements there. Still some challenges getting people to count and ID plankton, but we do have the samples and the water quality to go with it. And then lastly, I think what's really important is the work we're doing there with the weir and the assessment, I think complements the work of the assessment team, brings some efficiency to it. The people we have out doing the enhancement work are working together with those people. And overall, I think it's helping to increase engagement and capacity with our partners. So Tahltan Nation, and particularly in this watershed, Taku River Tlingit, and also the contractors we work with. Richard Earhart has been working with the TRT, and then Brian Mercer, who is really an amazing bush pilot, biologist, fish culturist, and a lot of work here as well. Yeah. Thanks very much, folks.
Great. Well, right down to the wire. Perfect. At 6:30, I wanted to extend, on behalf of the Northern Fund Committee, our thanks to Jeff, Flip, Aaron, and Corino for this snapshot of the many things that are going on in the Taku River watershed, in part enabled by the Northern Endowment Fund. The history, the scope, and the extent of collaboration between Alaska and Canada is truly impressive. I'd like to extend our thanks to your teams as well as the teams from Taku River Tlingit First Nation, as well as Tahltan Central Government. And then, with all sincerity, I want to extend our appreciation to the U.S. Department of Homeland Security and the Canada Border Services Agency, who have honestly facilitated the ability for personnel from Alaska and Canada to move back and forth across the border on an ongoing basis during the season, and then also facilitating some of our live egg and live fry transports every year. So it really is a multi-pronged effort across a number of Alaskan, federal, and Canadian, U.S. federal and Canadian agencies to enable these programs to occur. And oftentimes we hear about salmon no borders. Well, certainly the technicians and the biologists in the Taku River watershed know no borders as well. So that's really heartening to see. And then finally, to conclude, and we'll take off my Northern Fund Committee hat and put on my Fisheries and Oceans Canada hat. From a Canadian perspective, behind every good partnership is a strong leader. And on behalf of Canada, I'd like to specifically extend our appreciation to Alaska Department of Fish and Game Commissioner Doug Vincent Lang, and his consistent support, which has really enabled this unique program and the successful partnerships to remain a cornerstone of Taku River Pacific salmon management and assessment. So thank you very much, Commissioner Vincent Lang, and thank you all for your participation this evening. Oh, questions. Yes, we'll go to questions if there are any. Yes, go ahead. Run the microphone for those that are online so they can hear.
I had a question for the enhancement folks. So you mentioned that we've been doing this for 30 years of adult returns, fallen short of the production goal of 100,000. Cumulatively we hit 20 adult returns. I was wondering what the long-term average is in terms of production and contribution to bilateral harvest.
I don't know off the top of my head, but 10,000. There you go. Stock assessment guy, 10,000. 10,000.
In contribution to harvest?
Out and on a bit of 40%.
4,000 to 5,000 per year. Thanks.
100. Yeah, go ahead. Yeah, back.
This question is for Aaron. So after all these years working on this project, could you tell us what your favorite salmon species is and why?
Changing. It's always coho. It's a very easy question.
Good answer. Good answer.
All right. Okay. I think we're good for questions. So thank you again for your participation. I know it's late in the day, but do appreciate your interest, obviously in this showcase presentation, and then also specifically the work that's being undertaken in the Taku River watershed. So wishing you all a nice evening and thank you for your attendance.